Hello guys, and in this video we're going over the endogenous pathway of antigen presentation. There's the endogenous and the exogenous pathway. The exogenous pathway is going to be via MHC class 2. The endogenous is via MHC class 1 and will occur in all nucleated cells, Alright, and the exogenous pathway is going to be on antigen presenting cells such as your macrophages and your dendritic cells. So the first thing that happens is basically you're going to have a cell that, let's say, gets infected by a virus, and that viral DNA gets transcribed and translated, and it makes an aberrant protein, for instance, right? Or it could be a cancer cell that has an aberrant protein that's made. And what's going to happen is that protein is going to be tagged by something known as ubiquitin. Ubiquitin is a tag for an abnormal protein, and that protein will enter something known as a proteasome, which is the garbage can, basically, and is going to chop up the misfolded protein or the aberrant protein into little peptides. These little peptides will enter the endoplasmic reticulum via something known as TAP, and then it will be complexed to the MHC molecule, in particular MHC class 1, and that will be transported to the Golgi and eventually be placed onto the surface of the cell, where it can be identified by a cytotoxic T-cell or CD8 positive T-cell. The T-cell will identify that, and then it will cause apoptosis via the insertion of a pore known as perforin and the release of granzyme B. Perforin is going to disturb the osmotic gradient of the cell, so it can cause the cell to swell up and lyse, and the granzymes are going to basically induce apoptosis via the activation of the caspases. The caspases are a series of proteases that are inactive, so they're zymogens, but once they're cleaved, they become activated, and then they can activate subsequent caspases via something known as a caspase cascade. So one more time, and then once they're activated, obviously they'll chop up the cellular proteins and cause cell death. And once again, the perforin is going to disturb the osmotic gradient, cause the cell to lyse because of water entering the cell. Once again, you have your aberrant protein that gets a ubiquitin tag. It gets chopped up into the proteasome, into small peptides. Those peptides enter the endoplasmic reticulum via TAP, where they're complexed with the MHC molecule, and then they're transported to the cellular surface, where they can be identified by a cytotoxic T-cell. Natural killer cells also can identify aberrant proteins, except it's a little bit different. They're more general. They go around. They're innate, so they're not specific. Cytotoxic T cells are specific to a particular antigen, but NK cells will go around and look for, generally speaking, any cell type that has an aberrant protein that has MHC class 1 on it. Thank you for listening. If you have any questions, please send them in, and don't forget to please subscribe.